How do I know that it's actually started? Recording has started. Hello. Okay. I'm Cassie Bonvissuto. I'm the supervisor of the molecular diagnostics lab on the 5th floor. You guys will be spending like half a day in my lab with it's okay. It's okay. Is that 8 o'clock now? Is that now on the money? Yes, let's go. Okay.
You happen to know where sterile processing is?
Are you talking? Yeah, it's down the hall. That's where the autoclave and they clean everything. So if they keep going straight through this door, it'll be like the second to last door on the right.
Thank you.
You're welcome.
Oh.
Thank you. Okay. I forgot what I was saying. Oh, I'm the supervisor of the molecular lab on the 5th floor. I put in my 4-week notice, so Friday. So I won't be here to teach you next year, or potentially if you're going to be in my rotation. But you'll have half a day, and it'll be grand time in molecular. Hopefully, it won't be too chaotic. So yeah, I used to teach in my old life. I used to be a high school science teacher. I've worked in research. I've had a few different career paths. So I've been around. I've been around a lot. If you have any questions about potential like future career opportunities, just because you get your MLS certification doesn't mean that you're stuck in medical lab science. So know that. Use that to your advantage. Make yourself marketable. Yeah, this definitely pays more than teaching, so that's good. You made a better choice in that round. So it's not what I meant to. That's not what I meant when I asked you to rearrange the DNA. Right? He rearranged it from DNA to and. That's a cute little joke, but the first part of this whole lecture is review from high school, from your college biology classes, and hopefully from the video you watched for homework. How many of you, be honest, I'm not, Jen's not in here. How many of you actually watched the video for homework? Okay, we're making progress. That's like a 50%, we're good. That's okay. We're gonna speed through this, but if you need me to slow down, do not hesitate, but it's going to be a very long day. So I'm trying to not bore you with details that you may already know. So molecular biology, it refers to the branch of biology that deals with the structure and function of macromolecules essential to life. There's 4 major macromolecules, the ones that we really deal with in molecular biology in our lab. Are proteins and nucleic acids. Who remembers what the other 2 macromolecules are? Thanks. Yes, you guys are awesome. The molecules that carry out are DNA, RNA, and proteins. And this study has allowed for the development and implementation of diagnostic testing in the clinical lab. Because we can replicate DNA and we can manipulate it, and we can isolate it and tag it with fluorescent probes and things like that, we can do a lot of testing in the clinical lab in genetics and infectious. So what is molecular diagnostics itself? Is the detection and or analysis of nucleic acid molecules to provide clinical information. So do they have an infection? What is their genotype? We also do chimerism, which is engraftment studies. We check how much donor versus recipient DNA there is in their bone marrow and in their blood when they get a bone marrow transplant. So we can look at DNA for that. So we develop assays based on the structure and chemistry. And why do we do it? To search for molecular information to combine and complement other diagnostic information. Molecular biology cannot give us every piece of information. It's a combination. So let's say we do thrombophilia testing, genetic testing. We test for factor V Leiden mutations. We test for prothrombin mutations. We test for factor 13 and PAI. Just because somebody might have a mutation, let's say they're heterozygous, they have a normal copy and an abnormal copy, that does not mean physiologically that they are experiencing clotting disorders, right? There's multiple genes that work together to go through the coag cascade, right? And if you have one good copy, that doesn't necessarily impact its overall, like, your functionality. Some gene mutations have a higher impact than others. So you have to not only do the genetic testing, but you also have to do the coag testing, where you're actually checking the clot time in hematology. So it does not always provide the whole clinical picture. Again, it's all the tests that work together that provide the whole clinical picture. So we look at exogenous, so if you have a pathogen, and we look at endogenous, which is your own genetic DNA. So we do a lot of viral load monitoring in our lab for the hemonc kids. So a lot of the hematology oncology kids who are on chemotherapy and things, they are we monitor them for viral infections regularly, twice a week. Just because if they get one, it can go bad fast, right? Their immune system is shut down. So we're regularly monitoring them for even viral infections via like DNA, PCR. So like the viral DNA. So any questions so far? No? Okay. No? You just don't like me now? Okay, there we go. Outline. So these are things that we're going to go over in part one. Part one is the quick and nitty-gritty review. So we're going to go over molecular structure of DNA and RNA, the components and the key concepts, the central dogma. So that's DNA replication, transcription, and translation. Again, that should all be review. So this is a precursor to understanding the questions of molecular diagnostics, and we have to understand the DNA structure and function. So what is DNA? What does DNA stand for? Great job. Beautiful. Okay, so DNA consists of 5 carbon sugar, deoxyribose, a phosphate molecule, and a nitrogen base. There's 4 different nitrogen bases. There's adenine, cytosine, and thymine. So A, G, C, T. Who knows which ones bond to which? A to T, G to C. The apples in the tree, the cars in the garage. What about RNA? We're going to jump ahead. Who remembers what the 4 bases are in RNA? A, U, G, C. Good. And then A goes to U. Beautiful. You guys are so smart. Look at you. Okay. So we have our pentose sugar. The difference between RNA and DNA is this about right here. Oh, I'm blocking the board. Hold on. This is where the laser pointer comes in handy. Okay. So deoxyribose, right? You're missing an oxygen. Right. Okay. Your nitrogen bases, you have, they included uracil in here, right? In this picture. Okay, so you have C and T and U, which all have 1 ring. But you have A and G, which has 2 rings. That'll be important when we talk about bonding, right? A goes to T, G goes to C. We'll get more into the importance of that when we talk about DNA width. And yeah, so I always remember, this is what I used to teach my high school students. When we talk about purines and pyrimidines, I think that's a few slides away. But I like this, that this has a picture on this slide. So I'm going to kind of jump ahead. So do you guys know what purines and pyrimidines are? What are they? Okay, so pyrimidines have 2, purines have 1. So pure ag, like I think pure agriculture, AG, always has 2 rings. That's just a mnemonic that I remember. And then pyramids have 1 peak, right? So they have 1 ring. Yes?
I thought the pyrimidines were CT.
Pyramids, right? Oh. They have 1 peak, so 1 ring. Yes?
I thought you were putting an A and G.
Purids, pure ag. Sorry, if my laser pointer was off, I apologize. Did I say it wrong?
Someone did.
Did I? Listen, it's early. haven't been fully caffeinated yet. Pure ag, purines, pure agriculture, right? They have the 2 rings. And then pyrimidines, C and T. And then I always remember, I don't know why, but cats and tombs are in pyramids. I don't know, like, you know, Egypt. I don't know. Listen, like, that's how I just taught to my students back in the day. If it doesn't help you, it doesn't help you. And it might not be important. You might not even need to remember it. But that's how I did. So what is a nucleotide versus a nucleoside? A nucleoside is a base plus the 5-carbon sugar at the 2' site. It's referred to as unphosphorylated. And a nucleotide is that same nucleoside with a phosphate at the 5' end. So it can be a mono, di, or triphosphate. Do you know? What's an example of a triphosphate? ATP. That's a lot of people are like, what? Right? Because it's not in DNA, and they like pretty much associate like DNA and RNA to nucleotides, but actually ATP is also a type of nucleotide. Great job. So carbon positioning. A nitrogen-based carbons are numbered 1 through 9. Sugar carbons are numbered 1' to 5', and then the phosphate group on the 5' carbon. So it goes around in a circle. You can barely see it in this picture, but it starts 1, 2, 3, 4, 5. Okay, this is an oxygen group up here. That's not a carbon. So when we talk about these 1' to 5', it's actually this extra carbon out here that's number 5. So the phosphate group comes off this 5' end. And then the hydroxyl group is on this 3' end. And then you got that right. Here's the 4 nucleotides we went over. Oh, there's another picture of purines and pyrimidines, right? We got our purines and we got our pyrimidines. Again, this is the whole thing before. I just showed you the group, but now we have the whole nucleotide. Any questions? When we set up PCR reaction mixes, right, where we're trying to amplify the DNA, we put in DGMPs, DDMPs, DCMPs. We put in the individual nucleotides, right? They all go into a reaction mix because when we're replicating the DNA, you need a bunch of these extra pieces floating around so that it can actually be incorporated to make that new DNA. So that is part of what we call the master mix. So we put a bunch of them in there. So purines, double rings, pyrimidines, single rings. Here's where I wanted to talk about a purine always bonds to a pyrimidine because it's always 3 rings across. You want to keep the width of DNA consistent, right? You don't want to have a single ring bonded to another single ring, right? Because it's going to get skinny, and you can't have a double ring bonded to another double ring because it's going to be fat. You're not going to have a consistent width of your DNA. So to make sure that you have a consistent width, a pyrimidine always bonds to a purine. Now you can have mutations, and you can have misbonding, which does happen. But that alters the structure and the function and the ability of a gene to really and the DNA to really be stable. In PCR, I'm kind of jumping ahead again, but we really care about our CG ratio that's in the gene sequence. The CG ratio is how many C's and G's you have 10 nucleotides. If 6 of them are C's and G's, I have a 60% ratio, right? 60-40. Why would knowing the ratio of A's to T's and C's to G's matter?
CGs bind stronger.
They bind stronger. Why do CGs bind stronger? Because they have 3 bonds. Now, that's very important when we talk about our PCR cycles, because we're essentially breaking those bonds, heating that up, breaking the bonds, cooling it down, letting the probes and primers anneal, and then replicating and replicating. So a lot of things depend on these bonds. Temperatures, so we're breaking the bonds apart at. Hello, good morning. These things come into use when you're evaluating cycling conditions, and when you're evaluating probes and primers, and when you're evaluating things in PCR. So it's a very complicated process, but it works beautifully amazing when you have all of your parameters set properly. If you don't, you got to tweak some stuff. But if you go request a probe and a primer and things like that from, like, let's say Tidmol Bio, they will actually tell you the melt temperature based on the CG ratio. They'll tell you the things that you need to know to design your assay based on the sequence that you gave them. So technology has come so far since a few decades ago, where it was a lot of more hit or miss, and thermocyclers have gotten more consistent and things like that. So it's really great. Any questions on that so far? I know I'm kind of jumping around. I'm so sorry. So hydrogen bonds, again, guanine forms 3 hydrogen bonds with cysteine, and then adenine forms 2 hydrogen bonds with thymine. I always remember 2 between A and T because T stands for 2. C and G both rhyme with 3. Listen, bro, like, I used to have to teach high school students this. Mnemonics are my best friend. So these bonds are the key to the specificity of all nucleic acids. So based on test use, everything, again, is based on these rules, and that A always bonds to T and C always bonds to G, and there's always 3 bonds between C and G. Like, it's very, we depend on those rules and those actually working. So yeah, nucleic acid, DNA polymerase always adds to the 3' end to make synthesis occur. So we talk about synthesis in the 5' to 3' direction. Do you know that? Have you learned that yet? Yes? Okay, phosphate, okay, DNA polymerase, that's an enzyme, that's an ACE, right? It's building the molecule, adds to the 3' end. So you're always adding to that 3' end. If you look, it's a phosphodiester linkage. You learned about all those beautiful things, all the different linkages. It's a kind of bond that bonds amino acids together. Who remembers? Yeah? Yes. Look at you. You guys are so smart. I'm so proud of you. I'm throwing things at you that aren't even in this lecture. Great job. Yes, so you learned all those fun things in basic biology. You should have, but yes, a phosphodiester bond, okay, is what happens here. You get the phosphate group, bam, it bonds to the next set, and it makes the next nucleotide. So molecular structure, we have the double helix. Let me erase this 5' on the way. Okay, so we have double helix. Watson and Crick, even though you know there's controversy with that, but that's okay. You have your nitrogen bases. You have the sugar phosphate backbone, right? So your backbone of your DNA is those sugar phosphate molecules. If we go back, you can see here's the backbone: sugar, phosphate, sugar, phosphate. The nucleotides are the things, right, that are in the middle. Okay, so let's. Oh, okay. Also, phosphate is always at your 5' end, right? Phosphate 5. I don't know. That's another way that I remember things. They both start with pho. Again, I just a cheat that I used. Macromolecules made of nucleotides bound together by the phosphate and hydroxyl group on their sugars. So nucleic acid chains grow by the attachment of the 5' phosphate group to an incoming nucleotide to the 3' hydroxyl group of the last nucleotide. We went over that on the last slide. We refer to the DNA as oriented in the 5' to 3' direction. DNA in nature is mostly double-stranded. It is not always double-stranded. There is single-stranded DNA. There's also, most people think that RNA is single-stranded. There is double-stranded RNA also. So know that although it's a general consensus, that is not always the case, like for those rules to be followed. They exist in opposite orientation when they're double-stranded. So you have a 5' to 3' strand, and you have an anti-parallel strand, which is 3' to 5'. The sequences of the strands are complementary, which means, if you have A on this side, what do you have on this side? T, right? It's not identical. It's complementary, so it goes with its match. Again, I just said that they were anti-parallel in orientation, and identical sequences will not hybridize with each other. What does hybridize mean? Connect, right? They'll come together, they'll make that hydrogen bond, they'll sit nicely, they'll stick together, they'll be friends, right? Well, why won't identical sequences connect? Who knows? It doesn't work like that, okay. But why wouldn't it work like that? What did we just talk about? There's a lot of reasons, but if we go back to this picture can be helpful. So what did we talk about here? If we have T, what's going to happen if we have T here? What's going to change?
Too narrow.
It'll be too narrow, right? The width of the DNA. And then you'd have this other thing, a G combined to a G, and it'd be extra fat, and then we'd be narrow and fat and narrow, and it'd be like, woo, right? Although they do have the same number of bonds, right? It would be like willy-nilly, and it wouldn't be stable because those bonds wouldn't be able to necessarily get close enough to provide stability if there's fat ones in between and skinny ones, and it's just going to be a chaotic mix of events, and it's just not going to be happy. The double helix, okay, first described by Watson and Crick in 1953. We've come a long way. It's like only 70 years from when they first described the structure of DNA. In its hydrated state, in standard form, the beta form, it forms a right-handed helix. Now you can manipulate it to do other things based on charges and buffers and things that are in a reaction mix or in a physiological environment, but in their body under normal conditions and unmanipulated forms a right-handed helix. It's formed by those phosphodiester backbones of the two nucleic acid chains. There's about 10.5 pairs of nucleotides or base pairs per turn. So every time you go 360, there's about 10.5 nucleotides. Beta DNA, okay? This is the standard form. This is the form that you typically see in textbooks and everything. You have a very tight-ringed alpha form, and you have the zDNA form, which is expanded. You can't see it at the bottom, but you have the notes in front of you. I'll read it off my screen. So, beta, normal hybridization conditions, and it's a right-handed turn. And Z, again, high salt concentrations, magnesium chloride, or magnesium, the ion. Usually we add it to a solution in the form of magnesium chloride. That's what's in my brain. And it's a left-handed turn. So, again, you can manipulate it, but typically we work with the beta form. And then the alpha form is dehydrated conditions, and it's a right-handed form, or right-handed turn, and it's compressed. So, human DNA. Diploid. What's diploid? 2 copies, right? That means 2. We have 2 full complements of the genome. Each copy has about 3.2 billion nucleotides. We have 22 pairs of autosomes, which are our normal chromosomes, right? And then we have 1 pair of sex chromosomes. That's X and Y. Any questions on that? Overview. Beautiful. Okay, so here's where we get extra compact, and well, we're actually showing you the very most compact form is your chromosome. It's a very densely packed DNA. The chromosome is made of tightly wound chromatin. We have the nucleosome, which, hello, is your DNA wrapped around single protein histones, is 4 of those proteins put together, and then do it. And then we have the DNA helix, and then you have your nucleotide sequence. And it's a great time. Any questions? There's donuts if you came in late. Okay. RNA uses ribose as its sugar, and it has an additional OH. We talked about that. It's not deoxyribose, it's ribose, so it has that oxygen now. And again, that's in the 2' position. It's less stable than DNA. And the nitrogen bases, same, but uracil gets replaced instead of thymine. And it lacks that CH3 group of thymine, but it exists in double-stranded and single-stranded forms. A lot of viruses and stuff have double-stranded RNA. So that'll come in to account later when we talk about PCR and our thermocycling reactions. Wow, this picture is really atrocious. It's just showing you that CH3 group that is on thymine is now not on uracil. I apologize. I need to update that picture. That is awful. It's a little bit better on my screen. Maybe it's just, yeah. Oh, again, and also identifying the O, right, is missing in DNA and is present in RNA. Okay, so key concepts of DNA structure using molecular testing. Denaturation. Beautiful first step, right? You gotta separate those double strands. We can do that in a number of ways, or all 3, right? So you can have an alkaline pH. What's alkaline? Basic or acidic? Thank you. Strong hydrogen bond solvents, right? We want to dissolve those hydrogen bonds, or elevated temperatures. Elevated temperatures is what we use typically. We also have a change in pH, but PCR is mostly driven by elevated temperatures. What happens to molecules when you increase the temperature? Okay, wait, hold on. What did you say? They deform, but why? Because they're doing what? They have more energy. What are they doing when they have more energy? Well, they're just moving more, right? So are hydrogen bonds weak or strong overall? Weak. They're weak. So you're not breaking down the backbone of the DNA. You're just breaking the weak hydrogen bonds between the 2 strands. So they're getting faster. They're moving. They're like, oh, I have too much energy and I'm overcoming my bonds, right? And they're like, yes, I'm free. Okay, so we're good. We have melting temperature. So 50% of double-stranded DNA denatures to give a single strand, and depends on, again, we talked about this, the CG ratio, the length. The more nucleotides you have in your sequence, that could impact the temperature that you have, right? It's going to increase it potentially. The ionic strength and the pH of the solution. Again, depending on your pH, it might be easier or harder to break and denature those double bonds. And then the number of base pair mismatches. So again, we talked about it. If something's going wrong, right, and we got a mismatch and it's not bonding 100% correct, it's actually going to be easier to break that bond than if they're matched properly. So, the central dogma. This defines the relationship between DNA, RNA, and proteins in the transmission of genetic information. Okay, so we get DNA. We get replication when we make more DNA. Why do we do that? What process? Huh? Mitosis, meiosis. Mitosis. Copy our DNA. We make more copies. We divide. We make two new cells, and they're all happy, and they replicated, and we're great to go, right? In a cell. We're talking in a cell. Transcription and translation, right? That happens within a cell so that we can perform daily functions. So you transcribe your DNA to RNA. There's lots of different forms of RNA. And just FYI, throwing that out there, messenger is the most commonly talked about because that's the one that leaves the nucleus and then goes to the ribosome and gets translated into an amino acid sequence and makes your fancy proteins. There's lots of other RNAs out there though, so we'll talk about those later. But yeah, so transcription, you're rewriting something. This is what I tell my kids, right? If you're struggling remembering the difference between transcription and translation. Transcription, you're rewriting it. It's in the same language. You're just copying it, right? The only thing that changes is thymine to uracil. You're using nucleotides as your language. When you translate it, you're changing the language. You're translating it from English to Spanish. You're translating it from RNA or DNA, like our nucleotide sequence, to our amino acid sequence, right? Now, instead of nucleotides as our letters, we have amino acids as our letters. So that's how I help them remember the difference between transcription and translation. You're rewriting it versus you're writing a whole new language. So, replication, duplicates. We just talked about that. So you reproduce the DNA content from the parent to daughter cells, right? Mitosis. Oh, and meiosis. You get half. It's okay. You remember mitosis and meiosis? Sure. You don't remember?
No, I remember.
What's meiosis now? Yes, thank you. It's the making of your sex cells or your gametes, right? So it has half of the amount of DNA because then it meets with its other partner. You know, you do the nitty-gritty, meets with the other partner, and then you got a whole genome again. So meiosis, you copy it, but then you divide it in half. So you're replicating your whole genome, but then it still gets divided into half because you make your gametes, you make your sex cells. Replication, regular replication, the whole genome, just one copy, goes to your daughter, it's mitosis. Okay. Double-stranded DNA is made of single-stranded, and the sequence of the single strand dictates the sequence of the complementary strand. So replication is what we call semi-conservative, right? You have an original strand. Well, you have two original strands, and when you replicate it, you have your two original strands, but now you have two new strands. Semi-conservative. Each of those has an original, but each of them has a new. So somewhat conservative. Did everybody, were you able to see that? I was in your way. I'm sorry. Okay, so transcription. The process of generating RNA from the coding segments, right? So this happens in your nucleus, okay, where your DNA lives. You have your DNA template, it opens up, it says there's lots of enzymes involved, right? DNA polymerase, we got helicase. What does helicase do? Unzips your genes, right? Yes, good time. See, you guys remember all this. It's great stuff. And then you make a copy and we're good to go. And then this little single-stranded RNA will leave the nucleus, right? And then it'll go find its ribosome and it'll get translated, right? So translation, again, we're taking that nucleotide sequence and we're making it into an amino acid sequence. Yes. Oh yeah, it happens with codons, right? Our three nucleotides code for each amino acid. Yes. Part 2. This is where it gets exciting. Molecular technologies. So let me erase my semi-conservative DNA replication. So we introduced some methods for preparing and analyzing nucleic acids and methods for nucleic acid amplification, hybridization, and how to prevent contamination. DNA is everywhere. We have issues in our lab with DNA contamination in our assays. We'll talk about how we quality control with that. But if you think about it, if you flake a little skin cell on the table, your DNA is there. You'll literally just flake a little in your tube of somebody else. and your DNA is now in that too, with the patient saying, chaos can ensue. No, one skin cell, it's not even gonna really pick it up, but especially when they have so much DNA extracted too. But still, we have to be super diligent. So how the main things that we have to do, we have to prepare the nucleic acids for extraction, right? We're going to extract them from whatever sample we get, whether that be urine, plasma, whole blood, whatever. We will extract it. FFPE, what's FFPE? Anybody do like histo stuff? Formalin fixed paraffin embedded tissue. They come in little blocks. Have you seen them? No? Okay. Well, let's say they do a biopsy and they have a little tissue. They put it in paraffin. They fix it and they put it in a paraffin block, and then they can cut it and make very, very, very thin slides. And they can put it on slides and you can stain it for different things to see what kind of cancer it is or whatever, right? To see if it's infected with bacteria, to do whatever they need to do it. But we do also accept FFPE as a specimen type. It's a little bit harder to process because we have to break down that paraffin block. There's extra steps involved, but still, we have to extract it. Okay. Techniques for nucleic acid analysis. So like, how do we detect it once we extract it? How do we know what's in there? We do that. We have to amplify it. We use probes. And of course, again, we have to prevent contamination because of the problem. Okay. So the success of PCR depends on 4 things. A, specimen collection and transport. We'll tell you now. We have had specimens collected that we cannot isolate DNA from because maybe they didn't clean the area. They didn't let the alcohol dry enough, and they only gave us like 3 little drops of blood in a capillary tube, and now it's contaminated on their heel stick. They didn't let the alcohol dry. It's denatured and broke down all the cells, and we get crap. We don't get anything out of it. It's a great time. Again, also transport. If it gets too hot or too cold, like we can't accept frozen specimens for whole blood. We can accept frozen specimens for like urine and plasma and stuff. But when whole blood is frozen, the red cells break. It's chaotic, and we can, but we're not validated for that. DNA and RNA extraction and purification, right? How did that process go? What controls do we have to ensure that we got a proper extraction? We need to amplify the DNA properly and detect the DNA properly. Real-time PCR combines steps 3 and 4. So as we're amplifying, we're detecting. So as we're making more copies, we're watching a fluorescent probe or a chemical signal, right? Each time we go through an amplification cycle. So you'll get a sigmoidal curve. That's not going to show up. You'll get a curve that kind of looks like this, right? Because every time it amplifies, it doubles. So you start with 1 copy, you get 2, then you get 4, then you get 8. So you get the sigmoidal exponential curve until you run out of everything that you need to keep amplifying it. So then it flattens off, and you're like, this is your max signal you're going to get because I ran out of nucleotides, and I ran out of protein primers, and I ran out of everything that I needed. So now we're there. But each cycle, it takes a measurement. So there's types of analysis that you only measure the end product. You measure it once. You amplify, it goes through 45 cycles. You see how much is there after 45 cycles. Real-time PCR detects it in real time after every single cycle. So that's why it goes and combines step 3 and 4. Best and worst about PCR is the sensitivity. Again, if you accidentally contaminate something, you'll pick it up. Like, it'll show up. It'll be there, and you'll be like, that's not right. Right? And we gotta then go through a whole bunch of ways to clean and decontaminate, and it's a great time. Hopefully, the angle is to not contaminate it, right? It's very sensitive. It'll pick up, we can pick up 5 copies of an adenovirus in a patient's pee, right? 5 copies. That's like nothing. You start off with 5 little viruses. Boom, we can detect it. We can quantify it. We can do all that. It's a great time. But again, you shed a few skin cells in that sample. Anyways, it won't actually matter because we only have probes in that for the virus and not for human DNA, so it really doesn't matter. But like when we talk about like genetic studies, it doesn't matter. Anyways, but you can cross-contaminate between specimens, and that is an issue. We have kids who have billions of viral copies in 200 microliters of their urine. Just opening the tube can cause aerosolization, which can then get into other tubes in your hood. So you can easily contaminate other patient specimens. And I'll go into how we handle that later, but yes, contamination is a problem. Obtain useful samples of DNA free of proteins, salts, and detergents. Again, make sure when you're drawing somebody, the spot is dry after you've cleaned it. Your tubes are sterile. Your collection devices are sterile. You're collecting under absolutely the right conditions. We look after we've isolated it at the 260-280 ratio. The 260-280 ratio on a nanodrop instrument, which is a spectrophotometer, tells you how pure your DNA is. Is it contaminated with proteins? Is it contaminated with RNA? Did you not just get DNA out? Maybe you want RNA sometimes. We do just want RNA, right? So this 260-280 ratio is very important in the lab. Before we do any downstream processing, we nanodrop it. We make sure it's pure and it's what we want. If it's lower than 1.6, you're likely contaminated with proteins. If it's higher than 2.0, you're likely contaminated with RNA. Sometimes that doesn't matter, depending on what your downstream assay is, and other times it absolutely matters. So, you want to make sure that you do and you prepare your nucleic acid properly and you extract it properly. RNA is easily degraded by RNase enzymes, which are ubiquitous. You have them everywhere. They're more labile than DNA. What's labile mean? Liable. Yes. Guanidine isothiocyanate is a denaturing agent that inactivates RNases. We have this chemical in our lab that we use called DNA RNA away, right? It breaks down all the DNA and RNA, but we purchase DNase RNase free supplies because RNases, right? These are enzymes, they end in ase. They break down DNA and RNA. If we don't buy DNase RNase free tubes, and then we put our DNA in that tube, these enzymes are going to break it down, and then we can't use them for downstream processes. All of our pipette tips are DNase RNase free. All of our tubes are DNase RNase free. We have to put in a lot of extra thought and things when we're designing assays and purchasing supplies because it's so sensitive. Again, RNases are ubiquitous. They're everywhere. And we want a 260-280 ratio of 2.0 to 2.3. And if it's lower, it's likely contaminated with DNA, right? So if you absolutely are running an RNA assay, you want to get your DNA out of there. General DNA isolation procedures. Nucleated cells is what we get like genomic DNA from, right? And then viruses or bacterial, those are infectious agents. First, we have to lyse the cell. Again, we have to break it down, we got to open it, we got to expose all the DNA and RNA. It's a great time. So we either use detergents, we use lysis buffers that dissolve cell membranes and denature proteins. EDTA, chelase divalent cations required by nucleases, and then proteinase K degrades the proteins. So we always want to get rid of proteins. We use this proteinase K. We want to prevent nucleases from activating, right? Nucleases are what? They're going to do what? ACE enzyme, it's an enzyme, so what's it going to do? Okay, right? Okay, so we gotta open it up, but we also have to inactivate everything in it so we keep our DNA. So non-organic extractions, we precipitate proteins, and we end up precipitating DNA using alcohol. Did you ever do in high school or college, did you ever extract your own DNA with like, you spit in a cup and then you add like soap and alcohol and all this stuff? Did you ever like wind your DNA around a glass rod spool? Yes. Yes. That's fine. It's the same thing. They got lots of DNA. Yeah. So you've already gone through this process. How many of you have not ever done a lab to isolate DNA? Oh, I'm sorry. It's so fun. That makes my heart sad because I used to teach high school science. I feel like you should have done that in college too. What classes did you take? Did you take a general biology lab? How have you not done this? That's like basic biology. Where did you go?
I did mine online.
Okay. I will give you that. That is fair. I forgot. Okay. Where did you go?
Youngstown State.
You never did a lab?
I mean, it's been over
Okay, that's
10
fine.
years, so I'm like, I don't know.
I feel that, sister. I feel that. Okay. Specimen types. Most of the time we collect EDTA blood because if heparin and things like that, all the other great tubes out there that do lots of wonderful things for other collection processes, those inhibit PCR, right? We can only use EDTA. It helps us, it chelates the ions, we're good to go. It helps in that whole process. We can accept plasma, again, from an EDTA tube. Nasopharyngeal swabs, respiratory samples, cerebrospinal fluid, lesion swabs, urine, stool, BALs. We can like pretty much accept anything, but we're not validated for anything. The other day, I got a call from a doctor to run an HSV on vitreous fluid. What is vitreous fluid? From the inside of your eye. Gross. They had a viral infection on the inside of their eye. It was positive. It was a great time. I was gross. That's okay. They're getting treatment. That's all that matters. Great time. All these specimens should be stored at 2 to 8 degrees until they're extracted. Rapid extraction methods. That's what we use in our lab because who has time for manual extractions? We let cytogenetics do that one. When we used to do manual extractions pre-COVID, and then our lab got inundated like 900 COVID samples a day, and we were like, no, thank you. Somebody else can take manual extractions. So they did. So now we use all these cartridge-based extraction methods, and it's a great time. You don't get as much DNA. That's the downfall, but it's quick and it's easy. So upside. It's new and faster with minimal sample requirements, and it's magnetized glass beads that bind to DNA. We in the lab use the Maxwell RSC and use a QIAGEN EC1. These instruments offer various kits to extract various specimen types for genomic and infectious disease testing. We have a kit called the VTNA kit, which is a viral total nucleic acid kit. We use that for stools. Stools are also a gross specimen that require lots of extra pre-treatment because there are so many things in stools that can inhibit PCR, right? So we got to make sure all that extra gross is out of the way so that we can get pure DNA at the end. Plasma is so much cleaner. I mean, just look at the specimen types and you're like, no, thank you. Okay. It's a great time though. So this is what our Maxwell RSC looks like, and this is what our EZ1 looks like. This one can handle 14 samples at a time. Our Maxwell RSC 48 can handle 48 samples at a time. You just load each cartridge, you pop it in a row, you put an elution tube out front. That's the tube where your final extracted DNA will come into a nice little elution buffer. You put your sample in the back row into the lysis buffer that's in there. You pipette it up and down to well mix it. And then these magnetic bars come down with tips on them. And then they send it through the whole 7 cartridge process and they mix it and do the incubation for you so you don't have to do it. And it's so amazing. And then it uses these magnetic bead bars that attach to magnetic beads, and then attach to DNA, and it takes it and it spits it out in your final tube. And it's a grand old time. I love it. It's beautiful. So here's the demonstration on the process. You get your sample, tie it with your stuff. You lyse it, right? You get your magnetic glass particles, your MGPs. They bind the nucleic acid. So you can see the little magnetic particles are binding the nucleic acids. Then you have this magnetic bar that then magnetizes. You put in your sample and all your nucleic acid comes out. Leaves everything else behind, right? All the junk that doesn't have a magnetic bead bound to it, you put in the next one, you wash it, right? It's washed for additional purification. You repeat the wash, and then you get your purified nucleic acids. It's a great time. And all she had to do was put something in the first one. That's it. Techniques for nucleic acid analysis, we can have electrophoresis. Whoever ran a gel in school? Good, you've all run a gel. Perfect, love it. Okay, so this is electrophoresis. Enzymatic modifications, so color change. Has anybody ever run an ELISA in student lab? Yes, let's go. You get a color change, right? By an enzyme. And then endonuclease digestion, we can break it down, right? We use restriction enzymes to break it into smaller fragments. It separates in the gel. We can then determine all that good stuff. We band it out and then we tag it. What do you guys use? Well, I haven't run a gel in forever. I don't think they use a video bromide anymore because it's toxic.
We use.
You did? It's because it's cheap.
It's probably also partially it's toxic.
Yeah, it's also fair. As long as you wore the correct safety PPE, you are golden. No? Don't tell me that. I don't want to know.
This was in college and not with people DNA, so that's okay. We were
That doesn't make the chemical less toxic.
Yeah, I don't know.
It's a carcinogen. But if you use it, you're gonna, from one use, it's people who are running gels every day, like getting it all over themselves. Like what? You'll be fine. Anyways. Okay. I'm sidetracked now. Gel electrophoresis systems were the most frequently used method. They are not highly used now for molecular biology because they are outdated technology. It's great to run a gel as a double check confirmation method, but they're not used anymore, which is sad that, okay, so it is sad. It's great that they're still teaching you gel electrophoresis. It's an absolutely necessary tool. It's the foundation of molecular biology. But they need to step it up. When I taught high school, we had a thermocycler, we ran PCR, we did a lot of cool stuff. But I also had the experience because I worked in research before that, so I used a lot of my own experience to bring to the classroom. So regular high school biology teacher, I don't have that experience. That's okay though. In college, they should. Anyways, it's okay. You also were there during COVID. Maybe things will change. DNA is negative. Oh, movements of molecules by electric current, right? If you ran a gel, you understand that you have the positive end, you have the negative end, right? The DNA is negatively charged and will migrate towards the positive end. The bigger the molecules, will they move faster or slower? Slower. Great job. Okay. Again, so DNA migrates speeds inversely related to their size. So we also, for control purposes, run a molecular weight tracker, right? That has known sizes of DNA in it. And then you can determine the size of your DNA in the gel based on where it lines up with those known sizing markers. You always want to run control because there can be differences in gel consistency based on how well you dissolve the agarose. I've seen some people not dissolve the agarose very well before they pour the gel. It was clumpy, it just didn't run evenly, you get streaks. It's a great time, right? There's lots of things that can go wrong with gel electrophoresis. So it's not gold standard, but like it's not used anymore because there are so many things that can go wrong. But it uses an agarose matrix, so small pieces use more concentrated gels and large pieces use low concentrated gels. You control it. You control how much agarose you put in your gel depending on your size. If you are only looking for small molecules, you want more dense agarose to slow them down because they're small, they're gonna whip through the gel. The gels are only like this one, right? Like it's gonna whip through real fast. So you want to make it more dense. But if you're looking for large pieces, that is going to take forever to migrate through the gel if it's too dense. So you got to make it less dense. Lithium bromide. Here we go. An intercalase between the nitrogen bases, and you look at it under UV light. A gel can be run vertically or horizontally. It doesn't matter. So this is a test that we run in the lab. We used to run a gel for it. Now we do melt curve analysis, and that's okay. We have two normal patients. This is for Prader-Willi-Angelman syndrome. It's run with the same assay at the same time. We pre-treat the DNA by methylating it. And you get a methylated copy and an unmethylated copy. So they have different sizes. You have the paternal copy and you have the maternal copy. So the unmethylated one is larger. And the methylated one is smaller, right? So it goes further down the gel. Here's where it started. You can kind of see like the original loading thing. Every normal person should have a methylated copy and an unmethylated copy. You get the maternal copy and the paternal copy. If you have Prader-Willi syndrome, you're missing the unmethylated copy. If you have Angelman syndrome, you're I'm just speaking in circles at this point. If you're Prader-Willi, you only have the methylated copy. If you have Angelman's, you have the unmethylated copy. Okay. So we do this by melt curve analysis now, though, and I don't know if I included that in here, but I can pull an example for you after. It's a great time. They melt at two different temperatures. So you use probes and primers, you amplify the sequence, and you go through a melt curve analysis, and there'll be two peaks. If you're normal, you'll have both melt copy peaks. If you're abnormal, you'll be missing a melt copy peak. If you're missing both, you shouldn't be alive, so we wouldn't get your sample. Yeah. So electrophoresis buffers. So TAE and TBE. TAE is composed of tris, acetate, and EDTA. It's useful when recovery of DNA is desired, and it's used for large DNA fragments. TBE is composed of tris, borate, and EDTA. It's better resolution for small DNA fragments, and it decreases the migration of the DNA through the gel matrix. Again, we want to sometimes slow those very small fragments down because they can move too quickly. We don't want them to run off the gel. So, and obviously if they're small, they're going to be less visible, right? There's less DNA material there. So we want better resolution. That means separation, right? So it slows it down, but it separates it better. Enzymatic modifications. Polymerase, kinases, phosphatases, ligases, great times, right? DNA polymerase synthesizes DNA. Kinases, they phosphorylate the 5' terminus end of the DNA molecule. Phosphatases, they dephosphorylate the 5' end. And then ligases, you join strands, right? By forming those phosphodiester bonds. Did you learn about all of these? Like, these are all enzymes that, like, you know, should be familiar at least with ligases and phosphatases. And yes, no? Yeah? Okay, good. Oops. Okay. Endonuclease digestion. Restriction enzymes. They love restriction enzymes. They have a specific code that they go look for in the DNA, and then they go, I'm going to eat you and I'm going to cut you, right? Restriction enzymes are great for molecular biology methods, right? We can look for a specific gene by cutting it in places, running a gel, or we can, it's a lot for gels, but you can also use restriction enzymes to make new plasmids, right? And put genes into plasmids. What are plasmids? Do you know what plasmids are?
They're found in bacteria.
What are they though?
Extracellular DNA ring.
Did you just Google it? Okay. Yeah, it's a DNA ring. It's a little ring of DNA. They are commonly found in bacteria because that's how they pick up their little extra mutations that they're like, oh, I'm going to be antibiotic resistant this time. Let's go, right? They pick it up and they're like, yes, I picked up this gene from the environment. I'm going to stick it in my cytoplasm and make it. If it's useful, I'll keep it. If it's not, I'll get rid of it. Like, you know. So plasmids are little circles of DNA. We use plasmids a lot in research. You can use a restriction enzyme to make a cut in this plasmid. You can cut something. You can use multiple restriction enzymes. We can cut it in 2 places. We can cut it in 1. It doesn't matter. Anyways, once it's cut, we can then say, oh my God, I want to put a gene in here to make it exciting and new, like glow blue. And my teaching experience. I used to transfect bacteria with different plasmids with my students so they could see different colors and antibiotic resistance. If I wanted to put a gene for antibiotic resistance in here, I could cut it with the same restriction enzyme, mix it together, and because they have the same ends, right, it should insert right in there and be beautiful. And now I can transfect this plasmid into my cells, and then that bacteria or whatever will have that DNA gene mixed together. Okay, making new words today. And then it can be antibiotic resistant or be blue when it grows on a certain agar, or it can do whatever I want it to do. Plasmids are so amazing. I love them. Anyways, but a lot of restriction enzymes are used to run and cut your DNA so that you can run it and visualize it on a gel. But there are other applications for them as well. Ribonucleases digest deoxyribonucleases. Okay. Top 10 ways to prevent PCR. It's a good time. Change your gloves. If you have a specimen and you think you got any kind of specimen on your glove, change your glove. I don't care if it wastes a bajillion gloves a day. Change them. It's that important. You don't want to call somebody positive for something that they're not positive for because you didn't change your glove. It's terrible that way. Okay? Can openers for tubes. Cap openers. Wow, today. I'll finish caffeinating soon. Cap openers. So you're not actually messing with it. We have these little things sometimes, the little tools. Like, okay? So we don't have to physically touch it and potentially contaminate our gloves. But you should bleach the cap openers often, right? Between samples, you should bleach them. So what we do is we take 10% bleach on some gauze, we wipe them down. That way, because even the cap opener can transfer DNA from one tube to another tube, even though it's not you, it can still do it. Aliquot reagents do not go into the primary container that has 500 milliliters of whatever reagent that you need, and you only need 2 mils of it. No, aliquot it and then work out of the aliquot so you're not contaminating the huge primary container of reagent. Aliquot. Use barrier tips that have little filters in them. So they're pipette tips. You've all seen pipette tips, right? They have little filters. Okay, so that you cannot, if you aerosolize, if you pipette too quickly, right, you're aerosolizing, you have the potential to aerosolize the DNA. So that A, you don't contaminate your pipette, right? Or the air around you, or anything else like that, right? So you want a barrier. We still even bleach the pipettes though, because there's still potential. contamination. So we always clean, 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 clean. We have dedicated pipettes. We do not move pipettes from one location to another because as we go through the PCR workflow, you don't want your pipette that was dedicated for sample extraction to be in the same hood that you're isolating and making your master mix of probes and primers. You don't want specimen DNA to contaminate with your probes and primers, and you don't want that to contaminate anything downstream. So we have dedicated pipettes and dedicated hoods and dedicated brooms, and it's a great time. We keep them by serial number, so if they ever leave for service, we know where to put them back. We have unidirectional workflow also. So we have dedicated rooms for different things. So we have a dirty room, that's the main lab. Main lab's the dirty room. We get our specimen samples in here. This is our primary spot that we work. We do extraction in this space. So we have the dirty specimens and then the final DNA eluate, right? We get our DNA out. We go across the hall, okay? PCR1 is our cleanest space. No specimen, actually we don't take our specimens across the hall. Hold on. We take our specimens straight to PCR2 and leave them there. No specimen goes through this space. This is our cleanest space. This is where we add our probes and primers. We make our master mix. There is no foreign specimen DNA in this room. Clean. This is our clean room. Okay. PCR2 is where we take that master mix. Okay. We take it from PCR1 to PCR2, and then we add our specimen DNA to it. Okay. And then we go to the detection room. That's where we put it on the rotor gene, run a gel, do PCR, right? You never want to go this way. Never, 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 never. You have amplified your 10 copies of DNA to bajillion of copies. If you go backwards, what happens? Contamination. You take your samples and you throw them away. The amplified samples, not your primary samples. So those can go back in the lab for long-term storage, right? Like our primary extracted DNA that didn't get amplified can go back into the lab. Primary specimens can go back into the -70 storage for life in case we need them to pull them later for confirmation testing or whatever. But you never take amplified DNA product and go backwards with it. Never. Never, ever, 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 ever. Okay. Anyways, throw it away in the detection room. We have hoods with UV lights. Breaks down things, right? We use 10% bleach. We also use those DNA, RNA away solutions that break down the exogenous DNA and RNA. It's a great time. Do not use alcohol to decontaminate. We do use alcohol to clean, but we don't use it as a decontamination purpose. It doesn't necessarily break down the DNA. So yes, it can clean things, right? And it makes us feel better, but it doesn't break down DNA. It can kill like organisms, but it doesn't break down the DNA. Open tubes carefully, add template last and close the tube immediately. So when you are setting up your mix in PCR2, where you're adding your master mix first, you always add template DNA later. You don't want to go in with template DNA, add your master mix, and potentially spread that template DNA everywhere because you have now aerosolized it by adding something else to it. Right? You always add the master mix first, your template DNA last. Components in a reaction. Target DNA, you need it. Right? That's what you're looking for. Primers, critical. You need a primer. What's a primer? Anybody know?
It's like a small piece of DNA you add on to the template.
Yeah. So you have your DNA. I'm only going to draw one side of it. I'll draw two. Okay. You have primers. You have forward and reverse primers. That's what I wanted to draw here, right? We denatured our DNA. You have a forward primer that binds to one. You have a reverse primer that binds to the other. You're going to copy them, both strands, just of the gene region you want. So you're going to pick a sequence. You're going to blast it. What's blasting a sequence mean? Does anybody know? Blast. It's a you can use a computer website called NCBI. There's lots of other computer websites out there now, but NCBI was the first one. It's the main hub for all DNA sequences for every organism that is known, right? So let's say I want the CD4 receptor gene coding region for a mouse. I would blast the sequence. I would say, this is the region I want. It would give me a blast sequence for it. And I would pick a region that I want to then put my primers on. Okay? So, because then I'm going to copy that region. And the length of each cycle, right? In your PCR cycles, you set the time that you have like the replication occurring. Be 10 seconds, 20 seconds. It depends on how long you want this to be, right? The more nucleotides you add, the longer it's going to take for each cycle. So if it's a really short one, you might have a short cycle time. If it's a really long one, you want a long cycle time. But you need those. Those primers are critical. The magnesium chloride concentration is also critical. It can change how quickly and how accurately your replication or occurs, right? You need the polymerase enzyme, critical, right? You need your DNTPs. I talked about that. We add a whole boatload of DNTPs. Your adenine, your guanine, your cytosine, your thymine, right? You need those in there because we're making boatloads of copies. So it needs boatloads of stuff. We need buffers, we need tubes, water, and probes. Probes, what are probes?
Like a fluorescent.
Can't be fluorescent. As probes are, yes, it's a tag. It's a DNA sequence with a tag, right? It should be in your gene region that you're looking for, right? You want to tag that gene. You want to make sure that gene's there. So yeah, it's a fluorescently labeled, in our case, tag. We don't do other things besides dye labeled tags in our lab, but there are other ways. You can have enzyme labeled tags. You can have other things. Enzyme labeled tags, you would then put a chemical in there. The enzyme would digest it. It would change colors. But we use lights, right? It hits a fluorescent probe, is excited by one wavelength, and then emits another. You familiar with that? No? We'll go into that later. So. Okay, so this is an example of how the magnesium chloride concentration can affect your PCR reaction, your melt curve, things like that. When we were validating the factor V Leiden DNA assay, we looked at the different magnesium chloride concentrations. You can see that we have better amplification of our genes when we have 4.0 millimolars of magnesium chloride in there, versus like this. I think the red is 2, sorry, the red is 2, but you have green. It didn't work very well at 1, so it does matter. You want those probes and primers to bond correctly. You need the right ion concentration to happen in order for that to occur. So yeah, 1 didn't work at all. 2 worked a little bit. 2.5 worked a little bit better. 3 worked a little bit better, and 4 gave us the best amplification. So we want to use 4 millimoles of magnesium chloride. But if you go too far, again, it can have the same effect and inhibit amplification. So PCR, what is it? Again, you have your original DNA, you have your primers. We talked about that. That's critical, right? Even if you don't have a probe, because not every assay needs a probe. If you're running a gel, you just stain the whole thing with lithium bromide. That's not necessarily a probe. It can act as a final detection method. It's not what we call like necessarily a probe. So you have your primers, you have your nucleotides that we add a bunch of, right? And you have your original DNA. What's this process called when it separates? Somebody tell me. Denaturation. What's happening here? What do we call this when they hybridize? What's the other term? What's this called? Where we get the Okay, so those are great. Okay, so you get lots of copies. And it's exponential. We talked about that already. Okay, so PCR duplicates in vivo. Vivo means what? In the body. I thought you said, did you say in the bottle or in the body? Okay, I was like, not in the bottle. T, tube, right? Vitro, tube, T. Anyways, duplicates in vivo replication of DNA in vitro with the same results in less time. Okay, because we're controlling all of the fancy things in a controlled environment, so we can expedite processes. Again, you get this logarithm-based curve. It would continue to be exponential if we didn't have limiting factors. What are the limiting factors that we talked about earlier? Primers and probes, DNTPs, right? There are limiting factors. If there was no limiting factors, we could just build on forever and ever and ever. Stop copying. Okay. PCR modifications. So real-time PCR and RT-PCR. Okay. RT-PCR this is confusing sometimes because RT-PCR can stand for real-time, but it can also stand for reverse transcription PCR. It's dumb. They use it interchangeably, but technically real-time is also called qPCR. It's really dumb. When I first worked in research, I was confused by the whole thing that people do use them interchangeably at times. And I'm like, this is so annoying. But after you read the methodology of the paper, you pretty much find out if your starting material was RNA. We have to, if your starting material is RNA, you have to reverse transcribe it to DNA, right? It has to go to DNA, and then we can process. We currently in our molecular lab no longer have any RNA assays that we run all by ourselves. The COVID is now on a completely automated platform. We don't have to manually do that step at all. It's a great time. But it does have to go through that. But you have to reverse transcribe it, and it converts RNA into complementary double-stranded DNA, cDNA, so we can proceed with the PCR method. The reverse transcription reaction includes an initial incubation of 45 to 50 degrees C for 15 to 60 minutes. That is so that your first strand can be fully synthesized, depending on the length of it, right? You want to transcribe, you want to make the full thing of DNA. You want to start with good, solid starting material. The reverse transcription enzyme activity is inactivated by the first denaturation step. So, do you know why we can still proceed with PCR with DNA? Why would the high temperature not inactivate what we use is called Taq polymerase? Yes?
Is it because it's from something that's thermostable?
Yes, it is. Where did they isolate and find the initial thermostable enzyme that we? I'm sorry, what?
Is it like a thermophile?
Yeah, from like a hot spring. I think they isolated a bacteria from a hot spring. They got the enzyme. They were like, dude, let's go. They did great things. I'm not gonna lie. We'll probably never be smart enough to think about that in the future. Like in the long run, not there, right? Great people do great things. I'm not one of them. It's okay. And I'm okay with where I am. But it helps me now, right? It does my job for me. But the regular reverse transcription enzyme is not thermostable. It does not come from a bacteria in a hot spring. It's just going to be gone at that point. After you heat it up too much, it's like, see ya. So we have hot start polymerase, and then the PCR continues as normal. So qPCR. Amplification and detection occurs simultaneously. You can have real-time PCR that goes through reverse transcriptase first, right? You can have RNA that then goes through real-time PCR. Anyways, so you get amplification and detection that occur simultaneously. We talked about that. It requires a special thermocycler capable of monitoring fluorescence. So it will, after every cycle, it will capture the light that's coming off of it. You must have an addition of a probe that is specific or non-specific. We have lots of different probes. I used to use cyber green all the time in research. We don't really use that here. We use a little bit of it. We have hydrolysis probes, and we have hybridization probes, which is fluorescent resonant technology. And we generate amplification curves. It's a great time, and I'll go through each of these probes, I think in another lecture, not this one. Maybe it's this one. Okay, maybe. We'll see. I've read so many lectures at this point, they all like mush together. Okay. They use dyes to measure product accumulation. Probes increase specificity, but only yielding fluorescence when they hybridize to the target sequence. There are lots of different kinds of probes that work very differently, and we will go over that. That is in another lecture. There are several variations to the technique, and several methods can be combined. We can multiplex assays in a single tube. If it's labeled with a different color, we can multiplex it. One target could be labeled with yellow, another target could be labeled with green, and we can multiplex these assays. Growing numbers have been devised for a variety of applications. So cyber green is non-sequence specific. It binds to the double-stranded DNA product as it gets incorporated. So as it gets incorporated, as we're replicating this cyber green, it's not specific. It'll incorporate into whatever's replicated. So if the primers bind non-specifically to a different region in your DNA, and it amplifies that, you're going to detect it, right? But the great thing is that if it accidentally does that, it shouldn't necessarily melt in the same exact region and peak temperature as the one that you're looking for. We have two assays that use cyber green, HLA-B27 and the Prader-Willi-Angelman syndrome. We have a hydrolysis probe. That's our TACman probe, and the signal is generated when TAC extends the primers and digests the probe, which releases the reporter from the quencher. A quencher quenches the signal, so it keeps it silent, essentially. And the reporter is the actual fluorescent tag on it. So when we have hydrolysis, we have primers, right, on both ends. We have probes, and then the probe should bind where the primer is then going to come, and TAC polymerase is going to come extend, and it'll digest this whole sequence and replace it with nucleotides from the reaction mix. And when this reporter gets released from the quencher, it can be detected because now it's not being quenched or silenced by the quencher. Anyways, we'll quickly go through it. Utilizes two probes. Yes. No. So fluorescence, a FRET probe, it uses two probes. You have a detector, detection and the receptor. So you have these two probes, and when they come close together, they fluoresce. So our thrombophilia testing utilizes this. Again, primarily used in our genetic testing. Now, so we have these. We melt them off, and we do melt curve analysis on our thrombo testing. And when we melt them off, that we see a decrease in fluorescence. So we can look at the two different gene regions, right? Factor V Leiden mutation has a different melt temperature than the normal factor V Leiden gene. So if you have the mutation, the probes will melt off in a different temperature region than the other ones, and we'll actually see a decrease in fluorescence when they melt off. Okay, so what we do infectious disease testing, we do molecular genetics, so we do Fragile X syndrome, Prader-Willi Angelman, thrombophilia panels, we do HLA-B27 typing, we do engraftment, so we do molecular oncology. Sorry, HLA-B27. We don't do JAK2 or FLT3 anymore. We got like 2 samples a year. I was like, no, we're getting this. This isn't worth my time. And we do engraftment testing. So, yeah. And we kind of talked about that initially when we looked at donors versus recipients. Infectious diseases. Example of one organism of how a molecular test evolves. I think this is just one where everything we already know. Again, we talked about blasting the sequence of choice. You want to make sure, we talked about NCBI, right? That's where we blast it. You want to make sure that you pick a region of a gene, that is ubiquitous to every subspecies of that, right? Think about, you guys know more about bacteria probably than viruses so far. So if you want to just identify any Staphylococcus bacteria, right? No matter what subspecies it is, you want to look for a gene that will be consistent across all Staphylococcus species. If you want to just identify if you have Staph aureus, then you look for a gene that is just specific to Staph aureus, right? So depending on what you want, you need to blast and find a gene region that has no crossover between other organisms that may also be detected in that same specimen. Like when we are looking at our viral load monitoring for these TMOC kids, includes CMV, cytomegalovirus, EBV, Epstein-Barr virus, BK virus. We do adenovirus, parvovirus. So like we look at lots of different viruses. We don't want to choose a region that is shared across those. We want specific results, but we need a region that is specific to all cytomegaloviruses. That's not highly mutatable, right? We don't want a highly variable region also. So we want one that is stable. So when you identify primers and regions to amplify, you need to make sure you pick ones that are not highly variable, but are also unique to your organism. And that can take some time and some research. So the sample will contain genomes from other normal flora in the patient. You don't want to pick those up. Accumulation of dsDNA called amplifier defines a positive test. So if it's there, it'll be detected. So qPCR, additional sets of primers for other targets, right? You have to check if there's emerging strains, right? As we go through the COVID-19 pandemic, right? There were tests that were initially developed that the target that they were looking for mutated. The virus mutated, therefore they couldn't pick it up or detect it anymore. So a lot of times, a lot of the new assays to detect COVID use at least 2 different gene targets in them. So that way, if one mutates, you have a backup for another, right? So that they're not constantly having to change the assay. And yeah, master mix. It contains all the ingredients of PCR, right? You can purchase individually or company. We have LDTs. We purchase all ours individually. We develop our own tests, and we have to do all that stuff for COVID. And Fragile X. That's also a kit. We run an internal control, so a QC. How do we know that the extraction worked? How do we know amplification worked? We put an internal control into each and every sample. That's an exogenous DNA when we're talking about infectious testing, right? That's exogenous DNA that we're putting in there. It's not anything specific to anybody. It's made by the company. It says, this is my sequence, and it'll have probes and primers in their master mix for that sequence. So if your extraction worked, you should be able to amplify and detect the internal control. If your PCR worked, you should be able to detect your internal control. If your internal control is positive and your sample is negative, at least you know that the extraction worked and the PCR worked. Your control amplified inside that sample. If your sample is positive, you might not have internal control that's picked up, because all the nucleotides and all the reagents were used to amplify your viral DNA. If your viral load is very low, you'll have both show up. If your viral load is 300 billion, right, your internal control is going to show up. I like you. There's no big, you're super positive for your viral DNA. It took up all the reagents. So there's nothing left for the internal control to use. We add the internal control in a low level amount so that you don't have an issue with the internal control using up all of the reagents if you have a low positive viral load. So we add the internal control in a low amount. Step 3, denature, annealing, and extension. Again, we heat to 97 for 2 minutes. That completely dissociates the strands. We go through 40 cycles of denaturation, annealing, and elongation. 10 seconds at 97 to denature, 30 seconds at 60 for annealing, and 30 seconds at 72 for extension. So primers seek out their target sequences and will anneal. This is adjusted so primers do not bind to nearly identical sequences. You can adjust the annealing temperature to make it be more specific. If there's no target for the primers, no DNA will be amplified. So probes included. Again, we mix our probes and our primers together, and the target DNA will be in the region between the 2 primers. You want it to be in the middle of where your 2 primers are going to amplify. So yeah, inside a reaction vial, you have all that DNA, but here's only your DNA segment of interest. It can be very small. Your probes and primers, 15, 20 nucleotides, they're not going to be super long. Again, same thing. Test analysis, analytic, infectious, we're still talking about infectious. The probe fluorescent intensity increases. The CT, or the crossing point, indicates a positive result. So a positive sample must be positive. Internal control must be present if the sample is negative. Again, not necessarily there if your sample is positive, because all the components could have been used up. But we always set a threshold. Okay? If we do not call something positive, even if we start to see sigmoidal amplification, until it crosses a set threshold. Why? Why would we want to wait to call something positive until it crosses a set threshold? It could be a false positive. There could be non-specific binding that does happen. It occurs, but it won't typically occur over and over and over and over again with every cycle. So sometimes you get these little like wonky doodle things that kind of like, I don't know, they just do some wonky doodle things. And you get things that don't cross, right? Typically we'll rerun those. Could potentially be a low positive that didn't cross, but we confirm the negative result before we release it. So we set a specific threshold because we don't want to call false positives. Here's an example of our run. So we have a very positive sample, HSV1 control. We have positive HSV1 patient. And then we have a low level positive patient. You can see, here's our threshold that we set at 0.2. You can see all the rest of these samples on the run were negative. If I had a sample that started to come up over here, right, that would potentially either be a super duper duper low level positive or non-specific. We would repeat that one, just in case. But yeah, and we determined the CT, the crossing threshold, right, during validation. So we'll take known positives, known negatives, we'll run them, and then we'll set our threshold based on those results, right? Right, we want to repeat. We want to know that the positives were positive and then the negatives are negative. Reports, so post-analytic. We'll result them in a computer system. The reports include notes. The primers used in this assay may not be distinguished. So we don't necessarily have to say like what our probes and primers are, but we do have to say maybe what gene region we're targeting, just so people know. Sometimes it depends. It depends on tests. So ASR will include a comment. The test was developed and its performance determined by Akron Children's Hospital. It's not been cleared or approved by the US Food and Drug Administration. The FDA has determined that such clearance or approval is not necessary. It's used for clinical purposes only. And again, that's a clear requirement, right? We have to follow lab requirements. If we're doing lab developed tests, we have to include a statement that says this is lab developed test. And this fancy little comment goes all at the end of our reports. Genetic testing. That was all infectious. Mutant genes. Okay, look at those genes. They got 3 pairs of legs. Okay, so genetic testing performed in our molecular diagnostics labs. We do factor V Leiden, prothrombin, plasminogen activator inhibitor, PAI, factor 13, which is the valine 34 leucine mutation. We do Prader-Willi Angelman. We don't do JAK2 anymore. I lied to you. I lied to you. HLA, Fragile X, and short tandem repeats. Thrombo testing. Mutations lead to increased risk of thrombosis in pregnancy, deep vein thrombosis, and other things. So it's good to know what genes you have. 2 probes, once they hybridize to the amplified target, come close together, resulting in that fluorescence resonance energy transfer, right? We talked about that. That's the FRET. So they have that energy, the fluoresce, when they melt off, you don't get that fluorescent energy resonance, and you see a decrease in signal. Again, that's a melt curve analysis. So here's our cycles. Here's our temperature. We go through these cycles. We say, oh, I'm going to heat, I'm going to cool. Here's what I'm going to measure. These little green dots are like, I'm going to take a picture now. I'm going to see what we got. I'm going to see how much fluorescence we have, right? Through every cycle. Again, real-time PCR. So this is what it looks like. We're not looking at the amplification curve here because we don't really care about that. Yes, we want to ensure that we get amplification, but if we have a melt curve, we know that we have amplification. This shows you the melt analysis. After all 45 cycles are done, okay, we then melt it off. We go through a melt cycle at the end. We're just slowly heats it up, okay? You see, this is the fluorescence intensity as we slowly heat it up. We get a dip, then it flattens off, and we get a dip again. Those match up with the inverse fluorescence intensity, and this is where we call the melting peaks, okay? So this is our heterozygous control. We always run a heterozygous control so that we know both melt peaks appeared. So this is a previous patient that we are now using as a control that we know had, what are we running? Factor 5? I don't know. Prothrombin, okay? You have a normal prothrombin gene and an abnormal prothrombin gene. One person is homozygous for the abnormal gene, and the other person is homozygous for the normal gene. You could see that there's only 1 peak in that patient and only 1 peak in this patient. Okay? So, the red peak, you have the mutant. The green peak is a normal, and then that's our control. And they have defined ranges. So we have controls and we say it has to melt within these 2 set ranges. If it doesn't, it could be a different mutation, which we add a comment that says, hey, we can't say that this is the prothrombin. Oh, we look for a specific nucleotide at position 201, right? Like we look for a very specific change, but if it doesn't melt exactly, let's say it melted over here, right? Doesn't match up with our normal het control. That doesn't mean, this just means that they have something else going on and we don't know what it is. It's not detected by our test. We can't identify it, but there is something different. So we make a custom report. So yeah, there are different mutations that we just can't pick up. And then what do you think they would go do? They got a random thing. I don't know what this is. What do you think the doctor should go do to that person then if they think that they're at risk for the clotting disorder? Okay, they can do collard tests, but what's another test that can tell them specifically what mutation they might have?
Oh, genomic sequencing.
Yes, gene sequencing. We're looking at bringing up gene sequencing in our lab through an Illumina platform. It's just not here yet. It's expensive. We're working on it. So yeah, they could go and get a sequencing test for that gene, and then they can identify the mutation. Yeah, heterozygous. Just a little uneven. Anyways, I'm corny. I'm sorry. Cystic fibrosis, we don't do in the lab anymore. We used to do it, then COVID happened. We just too much. Okay. It's an inherited disease caused by mutations in the gene on chromosome 7. Mutations in the gene that make up the protein called CFTR. So cystic fibrosis, transmembrane regulator, right? So it regulates the passing of ions and stuff in your lungs and other mucous membranes. There's over 1,800 mutations found. We used to test for like the 27 commonmost mutations. Now it's cheaper to just send them out for sequencing. There's so many mutations, right? And they have targeted gene therapy for some of them. So knowing the mutation can sometimes change the treatment plan. Oh, I lied. Not 27. My bad, 39. I wasn't here when we did that testing. I was trying to just remember off the top of my head. My bad. We used the Luminex tag CFTR assay. But yeah, there are lots of other assays that test for significantly more markers, 271. They have kits that test for over that. We've talked about bringing those in because we do have a cystic fibrosis clinic here. It would be nice if we ran cystic fibrosis testing again, but the method that we were using, the doctors just were like, this isn't enough information. We need to send it out. There's better technology nowadays. So yeah. So again, it uses beads or microspheres coated with 1,000 fluorochromes, microspheres, and 23 transmembrane conductase regulators and 4 variants or polymorphisms. And then 16 of the most common mutations. It's a great time. So 1 in every 2,500 newborns has some kind of CFTR mutation. It's a good time. So we have those microbeads, right? So your biotinylated primer, your specific gene fragments. Again, we get DNA isolated, we amplify the gene sequence. Then we have the biotinylated primers. Then it like multiplexes. Each one of these beads has a different capture sequence on it so that it can capture the different mutations. So depending on the color, depends on what sequence you had, right, of that mutation. And then boom, you get laser, you get detection. It's a great time. Prader-Willi Angelman. It's a great time. They're clinically distinct. And their imprinting disorders mapped to the chromosome 15 on the Q arm at positions 11.2 to 13. And both disorders can result from microdeletions, uniparental dystony, so like where you don't get a copy at all. So you can either have a deletion, you could just be missing a whole copy, or imprinting center defect. So like something is added. So the DNA bisulfite conversion followed by PCR with melt analysis is used to first test. That's the first test in the diagnosis of Prader-Willi Angelman. After a positive DNA test, it should be confirmed by fish. Who's fish? And raw fish yet? Did you lecture with Sue yet? Sue Arnold? Melanie Lemos? No? There's a guy. Okay. I would think that the senior tech or supervisor from Cyto would be the one talking to you about it. Was it Dr. Julius?
No,
I love Dr. Julius.
I mean, it was just like brief. It wasn't like in depth as I was used to. Director of Pathology.
That's Dr. Braun. He's my medical director. So Prader-Willi Angelman. Prader-Willi is caused by the absence of paternally contributed or unmethylated DNA. Children should be evaluated if they demonstrate. So what's neonates with hypotonia? What's that? Low muscle tone. Okay. Mild prenatal growth retardation. Hyporeflexia and poor feeding. So if they don't have a lot of things, though, right? Could be. At 2 to 6 years old, again, still low muscle tone and a history of poor suck and global developmental delays. And then 6 to 12, history of poor suck, global developmental delays, and excessive eating with central obesity. So if you missed neonatal testing and you missed early childhood testing and you still got something wrong with you, you should probably get tested. Okay? Angelman syndrome is known as happy puppet. So we get a lot of tests. A lot is an overstatement. We get testing from newborns frequently if they suspect Angelman syndrome because they have very distinct facial features. So it's caused by the lack of the functional maternal copy or the methylated DNA. And it's the UBE3A gene, again, on the same chromosome in the same location as Prader-Willi. Again, one is lacking the paternal copy and one is lacking the maternal copy. The features include severe motor and intellectual retardation, ataxia, hypotonia, epilepsy, absence of speech, and unusual facial features, characterized by large mandible and opened mouth expression, revealing the tongue. So, again, this is another Melt curve analysis. We can see that, again, this is a decrease in fluorescence. Over time, you have one strict decrease in fluorescence, followed by a flat, and then another. Those are all the normal ones, the wild type. You have flat and then a decrease, which is Prader-Willi. Then you have a decrease, then a flat, which is Angelman. But again, if you put this on a temperature graph, right, you're increasing the temperature over time, and this is the inverse of fluorescence. You'll see a peak in this region. You can see how close all these controls are. These are the wild types. You can see that all these peaks are kind of right in the same area. And then you have your Prader-Willi and your Angelman peaks. Now, if you have one copy of something, you're going to have more of those nucleotides and DNTPs and everything is going to get used. So the signal is greater when you only have a single copy, right? Does that make sense? Here, we're splitting the nucleotides and all of the reagents between both sets of probes and primers and copies. So your signal is not going to be as great because it's getting divided by two things. Does that make sense? Yes? Okay. We don't do JAK2 anymore, but it's a point mutation substitution of guanidine to thymidine in the coding region of cytoplasmic tyrosine kinase. It's a great time. What's polycythemia vera? Did you guys wait? Did you guys learn that yet? Have you done hematology? Yeah, I didn't think that was. You'll learn it in hematology. You don't. I don't think you need to know that specifics for this. Essential in thrombocythemia and primary myelofibrosis. And again, it's Melt curve. Again, we don't do it anymore. HLA-B27 testing. So there's lots of human leukocyte antigens, right? When you go for a bone marrow transplant, right, they do HLA testing to make sure that your cell surface markers are matched with the person that you is potentially going to be your donor so that your immune system doesn't attack the foreign cells. This has nothing to do with it. Well, it does, but like it's only a piece of it. But it's common if you have ankylosing spondylosis. I don't know. Anyways, it's a chronic inflammatory disease caused by pain. I think that I spelled that wrong. I think I need to fix it. Anyways, causing pain and inflammation in the joints between the vertebrae of the spine and peripheral joints. 90% of HLA-B27 patients, 90% of patients who are diagnosed with this have HLA-B27. So it's not confirmatory. It just increases your likelihood of developing or being diagnosed with the disease. There's a high correlation, right? So it can also be positive in Reiter syndrome. with an inflammatory bowel disease, but again, is not diagnostic of these disorders. So people run the HLA-B27 test if they suspect something, and then that kind of like solidifies it. Oh, yes, they have this. Beautiful. Makes sense now, right? Most patients who have this also have this. It's kind of like confirmatory, but not. Fragment analysis. This is my favorite instrument, and we do sequencing. So this is our 3500. What? No, that's the next slide. I lied to you. I can see it on my screen. That was my 3500. I saw it on the right. So fragment analysis. We label fragments of interest with fluorescent dyes. We amplify the labeled fragments, and we separate the fragments with capillary electrophoresis. That's what this is. CE is capillary electrophoresis. Again, it's like gel electrophoresis, except significantly longer in these long capillary glass tubes. They could be different lengths. I think the length that we use right now is 55 centimeters. I don't know. I ordered it. I installed it. I don't remember how long it is. I think it's like 50 centimeters. I think that's wrong. I don't know. It doesn't matter. Anyways, we size it using a standard. Again, just like gel electrophoresis, you put something through the capillary array that has a known size, right? Capillary electrophoresis, the sizes are, it goes through the capillary electrophoresis based on size and charge, right? Again, larger molecules are going to move slower. We can also genotype using software that assigns an allele called based on different loci. This is sequencing. I'll show you. We don't do sequencing on our guy. I want to. We don't yet. I'm trying to bring in 16S sequencing for bacteria. But you determine the base pair sequence of DNA fragments by the formation of extension products of various lengths. So sequencing works, the basic sequencing works, like you have a bunch of nucleotides in your mix, but there's like a portion of them that are fluorescently tagged. Not every single nucleotide, when you're doing sequencing, is not fluorescently labeled. If there is a fluorescently labeled one that gets incorporated as you're PCRing, like let's say those two aren't, but I get to one that is, that's fluorescently labeled, it will not add another nucleotide, it will stop. Okay, so then you get different length fragments based on when a fluorescent nucleotide gets labeled. And then you can sequence it and run it through the capillary array, and it'll tell you, actually it'll fluoresce because it's based on size. If I only got to 4 this time, right, that's going to be a whatever color blue is. G, I don't know. And then let's say this one is A, right? And that'll be in position, that'll come in position 1, 2, 3, 4, 5, 6, 7, right? Okay, we know the 7th position is A. And you'll run it through, and then you'll eventually get all of the different sizes because it randomly incorporates. So you will get, after so many cycles of PCR, you will get something that randomly stops at every single spot that gets incorporated through all of those cycles. So you'll get the sequence. It's a great time. That's our doohickey. That's our instrument. So it utilizes the separation of ionic fragments by size. Negatively charged extension products of PCR enter the capillary as a result of electrokinetic injection. So there is no displacement of matter, right? Yes, you'll lose the particle into the capillary array, but it's not like injecting like 10 microliters of a liquid, right? You're just getting the molecule. There's no liquid that's coming with it from the solution. So you get electrophoretic mobility affected by run conditions. Again, pH, buffers, temperature, voltage. You know that even changing the temperature in the room while this thing is running, because it can run for hours and hours, can actually affect how fast your molecules move through the capillary array. It can mess up your data. It's a great time. And by great time, it's annoying. because our lab is in the oldest wing of the hospital. And old buildings, old parts of buildings, they're not the best temperature regulated. It's okay though. We work with what we got. Injection methods. So, hydrodynamic injections is where you get the fluid with it. Hydrodynamic, there's fluid with it, right? In a capillary array, you're only getting a charge, right? It's only the molecules. There's not really fluid that brings it up. So, this is our capillary array. So these are the nice little glass tubes, and they curve around, and they go through this detector box. It's covered in black because that's where the laser lives, and you don't want the laser to just be shining out into everybody's life. And they're very thin, and they're glass. Could break them. When you install them, those are hundreds of dollars to just install a capillary array. But yeah, it's my favorite instrument. The grand time. Personal love detector box. This is where all 8 capillaries come in through this little tiny visual field, and the laser comes through and excites it, and then it goes, oh, there's blue in this capillary, and there's nothing, and then there's orange, and then there's green, and as the colored molecules pass through, it's like, there I am, and then it's good. So what does a CCD camera see? So each dye emits a light at a different wavelength, and then it'll put it on a nice little graph for us. I didn't show you one of those, but I think there's one coming. So Fragile X. So our fragment analysis we use for Fragile X, short tandem repeats, and flip 3. Here's Fragile X syndrome. It's the most common cause of inherited mental retardation. It affects about 1 in 4,000 males and 1 in 8,000 females. Why would it affect more males than females?
They don't have one else.
We got 1 X, for sure. Okay. Males only have 1, so if they get a messed up copy, there you go. You're SOL. Females, we got to back up because we're better. I'm just kidding. You're right. In most cases, how many boys are in here? Just you know, it's okay. In most cases, it's caused by the expansion of a random CGG repeat. And it's at the end of the FMR1 gene on the X chromosome. Normal people, we have 5 to 44 repeats in the CGG repeat. That's normal. There are many places in your genome where you just have random repeats, right? A premutation is 55. Well, that is not right. Oh, okay, yeah. 55 to 200 repeats. Unstable gene, it's an unstable gene if you're a premutation and can expand in the next generation. You likely will not see any effect if you have a premutation, unless you can have some symptoms if you're a boy, which is like you get tremor ataxia, and in females you can potentially get premature ovarian failure, but again, that's not, it's just a higher likelihood that you will. So again, it's not 100% that you'll get either of these, but because it's already unstable, it's already too large, you might have these symptoms and your likelihood of passing it on is going to be even greater because it's already unstable. A full mutation is greater than 200 repeats. And then that causes silencing of the FMR1 gene. It's too big. It can't do what it needs to do. And then you get a lot of different symptoms. Boys have, again, mental retardation. Women, you might want to do family studies. You might want to get, like, you don't want to pass that copy down. Like, you might not necessarily still have symptoms, like you can, but you have an extra copy, so you're not necessarily going to experience them all. So Fragile X, a normal person, this is 19 CGG repeats. Here's our control. Again, we want to make sure our assay is detecting all of the possible regions so that we know our capillary array is working right and spacing properly. So we run a control that has lots of different repeats. We have 29, 160. We cannot characterize anything greater than 200. So we just say, oh, it's greater than that. You got a full mutation. There are labs that do characterize them. We do not. So we clump everything in the red category to greater than 200. And you get diagnosed with Fragile X syndrome. FLT3 is a member of the class 3 receptor tyrosine kinase family and is primarily expressed on hematopoietic stem cells. 20 to 35% of adults and 1 to 17% of infants and children with acute myeloid leukemia, AML, have one type of the FLT3 somatic mutation, which is an internal tandem duplication of the juxtamembrane domain, and it results in an increased uncontrolled self-proliferation, so of the leukemia cells. Again, if you have FLT3, that is not good, right? It says that you have a poor prognosis. Yeah, it's really sad. But we used to do that testing. Again, we only got like 2 years, so we set it up. But, yeah, so a FLT3 positive patient, you have your normal, then you have your positive ITD mutation. Negative, you have your normal but nothing. Your positive control, you always want to make your controls low positive. That way you can make sure that you're detecting any low positives. So you look at the ratio between your normal and what you get as your positive result to determine the ratio of expression. And your negative control should only have your normal. Microsatellite testing or short tandem repeat. This is what we do for pre-transplant, post-transplant bone marrow engraftments for kids. We look at the percent donor and the percent recipient. We can also run it on maternal contamination to rule out maternal blood for fetal genetic testing. So we'll look at that and we'll say, okay, this blood is good to go for any other genetic testing because we can say that there's no maternal DNA in it. And this blood isn't good because there is maternal DNA and contaminating it. So you might not get the results you want. You will have to take a different sample. We can also do twin zygosity analysis. If twins are identical, they'll have all of the exact same 27 repeat areas. And if they're not identical, they won't. So even if there's only one difference, they're not identical. So there you go. This is the same testing that they do for like DNA fingerprinting, right? It looks at random repeats. So if there's where the 27 came in. Oh, 39, CFTR, 27. Here we go. I knew 27 was important. Engraftment or chimerism analysis is used to determine the presence of recipient or donor in hematopoietic cells. Donor or recipient differentiated by capillary electrophoresis using 27 loci. So the likelihood of you sharing all the same repeat numbers in these random areas across 27 different loci, it's probably slim to none, right? I'll show you. So we get a DNA profile is produced by separating DNA molecules by size and dye color. The labeled fragments are separated based on size. They're not detected on the gel. We just do capillary electrophoresis. It takes about 2 hours to run through the instrument. You get an initial peak layout of all the colors, and it'll separate it by dye color on a different graph. But you can see that, okay, for this red color, this will be one loci. You have repeat number 5 and 7. I'll show you in a little bit here. Oh, no, I don't have a better picture. I do. I don't know why. Oh, yes. So these are the spots that we look at. TPOX, we look at those genes and the repeats in those gene regions. Some of them are named based on their chromosome location. Okay, this is on chromosome 5, S818, that's the position. This is chromosome 8, S1179. So some of them have actual like names. Some of them are just named by what chromosome they're on and their location. But we look at 13 of them and their chromosomal positions. So this is one loci. This patient has maybe on one chromosome they have 14 repeats, and on the second chromosome they have 17 repeats. For this loci, both of their chromosomes, they have 6 repeats. So if there's only one set, like if you have double the amount, like same amount, number 6 from mom, number 6 from dad, there's more DNA for that. So you'll get one taller peak, right? If you have 2 different ones, again, your detection signal is going to be about equal for each peak because you have each copy. Does that make sense? 2 copies. So we can say, okay, patient A has 14 and 17, 6, 28, 31, 16, 19, 11, and 12. Patient B has 14, 17, 6, 28, 31, 16, 19, and 11. Are they the same? Why? What's different? Okay. Number 12. What else did you say?
I said 12.
Oh, I thought somebody said 13. I was like, oh, 12. Yes, 12 in the penta-E region. Okay. Yes. So they're not identical. Beautiful. Great. So pre-transplant. My recipient has this. My donor has this. My post-transplant, I have a mix of recipient and donor. Looks like I have a little bit more donor than I do recipient though, which is good, right? We can actually calculate the percent recipient and donor by calculating the peak, the area under the peak. So if there's an area of 200 and there's an area of 100 under this peak, right? We can calculate the donor area divided by the total area and get percent donor. We can also do it the other way. Recipient area divided by the total area to get the percent recipient. Or if you calculate one, you do 100 minus the other. That makes sense. So this donor or this recipient has this peak and this peak. The donor has this peak and this peak. We can't use this peak. It's shared. So when we can still use it to do analysis, we would just use these 2 peaks only. We can't use the shared peak. So we use D1 if we're calculating donor divided by D1 plus R. That should say 2, but it doesn't. But it should. But yeah. So there's lots of different ways that you can do it. You can have 2 different ones. You can't really use this one at all. We shouldn't use because they share one. So you wouldn't use that one. But we know it's different. So new technology since the pandemic. This is already shut down. We bought this like $500,000 instrument to run like 800 COVIDs a day. We don't do that anymore. So it's a big waste of space. But it was a great investment in the time, and our techs greatly appreciated it. They didn't have to hand pipe a bajillion samples a day, and it was a great time. And that was pre-me for that decision. But it is now shut down. We have the mnemonics. The reason this is shut down is because it runs everything in batches of 96. We don't get things, we get like 2 COVIDs a day now. This is not feasible or economical to run this instrument. This can also run 96 at a time, but it's single load. So you can load one as you get one, and it's good. It's all test strip based cartridges. You're not wasting all 96. It'll pierce a cartridge as it needs it. So it's a great thing. This was pre these 2. It also ran 96 at a time, but there was we still had a lot of manual processes. All this would do would be like pipette everything into the wells for you. And then we still had to then go add the master. Like we had to do a lot of other things and put it on the thermocycler and analyze the data and resolve the data. These 2 were interfaced. I got the result, crossed it over to the soft, and also you can say, verify. It was a great time. Before then is it was a lot. It was a lot. Testing considerations. We've added adenovirus quants. We added the 4-plex, we validated it, and then they stopped producing the test strips. Because they didn't meet the qualifications when they got rid of the emergency use authorization. They didn't meet the qualifications for complete FDA approval. They were going for FDA approval on their 2.0 version of this, which wasn't going to be right. They were focusing all their eggs in the basket to get FDA approval for their 2nd version of this, so they discontinued this. We went live for like a day. It was a great time. It was a great time. We don't do that anymore. And then spinal muscular atrophy, SMA testing. We also don't do that. But yeah. Maybe we'll bring those back. It'll be a great time. Analyte specific reagents sold by companies. We use Altona, but we also do a lot of in-house validations. We will blast our own sequences and order them lyophilized from Tidmol, and we have to do lab testing, homebrews, right? Those are all our homebrews. They require significantly more validation associated with it than if you bought an IVD test from a company. They come with the validation kits, you run the 40 samples they give you, boom, it's done, right? We have to do a lot more validation because we're making our own. So we have to really prove to people, yes, this is legit. We're having our own internal control processes as we order probes and primers and validating each lot, whereas the company would validate their lots of probes and primers before they even shipped it to us, right? We have to go through extra validation each time we get a shipment of something and we order something. So, yeah. The FDA, you can get FDA approved assays or research use only assays. We can use RUO products and use them in our validation though. So, but we just have to validate it and provide all that evidence.